All right, let's jump right in. Picture this. You're an environmental detective. You're on a massive case, trying to figure out how to save the fish in our most important rivers. The problem is, the scene is huge, your targets are vanishing, and the absolute best clues? They're microscopic. They're invisible, and they're floating right past you. Today, we are cracking the code on how scientists are using some seriously cutting-edge DNA tech to read these tiny clues and hopefully save entire ecosystems.
And that really brings us to the central puzzle for conservationists everywhere. I mean, think about it. How in the world do you protect a species nursery when you can't even tell which babies belong to which parents? We're talking about thousands upon thousands of these tiny see-through fish eggs and larvae just drifting along in the current. They're the future of the entire river, but to you and me, they all look pretty much the same.
And this isn't just a small issue. It's a massive roadblock. See, if you want to protect a fish population, you have to protect where it lays its eggs, right? The spawning grounds. But if you can't tell the egg of an endangered species from a super common one, how do you know which river needs saving from a new dam? Or which area needs to be protected from pollution? The old school way of identifying this stuff, it's called Ichthyoplankton, by the way, is just painfully slow. It's difficult, and honestly, it often leads to really costly mistakes.
So, if our own eyes can't solve the mystery, we need a totally new tool. And this is where science has a major breakthrough, by cracking the genetic code that's hidden inside every single one of those tiny eggs.
This incredible technology is called DNA metabarcoding, and the idea behind it is just so cool. Think of it like a genetic barcode scanner at a supermarket checkout. Scientists can just take one single sample from the river, like a scoop of water that's full of dozens of different eggs and larvae, and this technique reads a specific little piece of DNA from everything in that sample, all at once. It's so revolutionary that scientists are calling it biomonitoring 2.0.
The process itself is actually pretty straightforward. First up, scientists go out and collect their samples. Step 2, they zoom in on that specific genetic barcode they want to read. Then, step 3, these super powerful sequencing machines read millions of these DNA barcodes simultaneously. And finally, this is where the real magic happens. Powerful software matches each one of those barcodes to a massive library of known species. And just like that, you get a complete list of who's in the river, where they are, and when they're spawning.
But here is where the plot thickens. This amazing tool, as powerful as it is, can be a bit of a double-edged sword. Because if you don't use it in exactly the right way, you don't get a river of clues, you get a river of complete misinformation.
And this leads us to a really critical question that some scientists down in Brazil decided to tackle. They were working in the San Francisco River Basin, and they asked a simple but profound question. Does how often we sample, how frequently we dip our nets in the water, does that completely change the story we think we're telling?
So they ran a simulation. And when they used a monthly sampling schedule, which is a pretty common way of doing things, the data pointed to one very clear conclusion. You can see it right here. The main branch of the San Francisco River, that SFB site, has a huge spike in egg density, so the logical takeaway is, aha, this is the spawning hotspot. This is the place we need to protect.
But here's the kicker. The research team also took samples every 3 days. And when they did that, whoa, the story completely flips on its head. With this high-frequency data, we suddenly see that the main river, it actually has a low density of eggs from the really important migratory fish. The real action, the critical spawning grounds, are actually in these two other rivers, the Bambuí and the Samburá. I mean, it's just a shocking example of how you could end up pouring all your resources into protecting the wrong place entirely.
And the stakes here could not be higher. With that less frequent monthly sampling, scientists would have totally missed the spawning of 4 of the 7 key migratory species. That means endangered fish, like the huge Suruban catfish, would have been completely invisible. Their nurseries would have been left totally unprotected, all because the timing was off.
Okay, so you're thinking, great, just sample more often. Problem solved, right? Well, not quite. The challenge doesn't stop there. It turns out it's not just when you look for clues, but also what tool you use to look. And this brings us to what we can call the scientist's dilemma. You can think of these different genetic barcodes, or markers, like different lenses on a camera. The old school one, called COI, it's kind of like your wide-angle lens. It has a massive library of known species, so it sees a lot, but sometimes it can't quite focus on some of the local fish. Then you have these newer markers, like 12S. These are more like a zoom lens. They're super sensitive for picking up certain native species, but their reference library is way smaller. Each lens, you see, has its own blind spots. And a 2023 study by Texera and its team put this to the test. They ran the exact same samples using different markers, and the results are crystal clear. Some species were only picked up by that COI wide-angle lens. A whole other group was only found by the 12S zoom lens. And then, yeah, there was a group that both of them saw. The takeaway here is impossible to ignore. If you use just one tool, you are going to miss things. Period.
And the impact of using multiple tools together is just staggering. I mean, the study itself puts it pretty bluntly. Combining the markers boosted their detection rate by up to 87.5%. That is not a small improvement. That's the difference between having a vague, blurry sketch of an ecosystem and having a detailed, high-definition blueprint. So, what does this all mean for the future? Well, it means that science is moving past the early days of this tech. We're now entering the next generation of biomonitoring, and this time, we're armed with a much smarter, more refined, and way more powerful approach. Scientists are now building this much smarter toolkit. It starts with combining multiple lenses to get that complete picture. They're also working like crazy to expand those DNA reference libraries so the barcodes actually have something to match against. They're even validating their methods with something called mock communities, basically samples where they already know what's inside, just to make sure the tech works. And of course, they're setting up clear rules for things like sampling frequency. This is how good science gets even better.
And all of this refinement, it's everything. Because of this hard work, we're no longer stuck with just a single, blurry snapshot of what's happening in our rivers. We are now actually capable of creating a high-definition movie, capturing the complex, dynamic, incredible story of these ecosystems as it unfolds second by second.
And that leaves us with this final thought. We are learning, for the first time, how to read a story that is literally written in the water all around us, a story told in strands of DNA. And as we get better and better at reading this incredibly intricate language, the question really becomes, what other secrets are out there, waiting for us to unlock? What other mysteries will we be able to solve?